Welcome to this explainer created specifically for St. Paul American School Clark Grade 9 Biology. Today, we're literally building a plant from the ground up. We'll start with the microscopic cells, build up to the macro architecture, and then we're going to explore those amazing environment-sensing hormones. So, let's get into it. Here's our agenda for today. First, plant cells and tissues. Second, roots, stems, and leaves. Third, plant hormones and responses. Then we'll do a quick misconception and formative check. And finally, wrap up with a concise recap. Section 1. Plant cells and tissues. The microscopic foundation. Okay, so the absolute foundation here is that plant tissue systems basically fall into two general categories. First up, we've got meristematic tissue. You can think of these as the plant's literal growth engines. They're found in areas where cells are continuously dividing, like the very tips of the stems and roots. The cells here are constantly splitting and totally undifferentiated. But on the flip side, we have permanent tissue. You see, once those meristematic cells differentiate and actually take on a specific job, like, say, protecting the plant or transporting water, they completely lose their ability to divide. They become permanent. A really brilliant example of complex permanent tissue is the vascular system. The contrast here is super fascinating. The xylem transports water and nutrients up from the roots, right? But what's wild is that its main conducting cells, like vessel elements, are actually dead at functional maturity. They literally just act like hollowed-out pipes. Phloem, on the other hand, is busy transporting organic compounds and sugars from the leaves to the rest of the plant. And those conducting cells, they are very much alive. Section 2. Roots, stems, and leaves. The macro architecture. Now, plants grow by rapidly dividing cells right at their tips. This is called primary growth, and it's what lets them stretch out to seek sunlight and water. But here's where it gets really interesting. Apical dominance. The apical bud right at the top of a shoot actually releases signals that suppress the growth of the lateral buds, the ones along the sides. That's exactly why coniferous trees, like your classic Christmas tree, have that totally distinct conical shape. If a gardener were to clip off that top apical bud, the side branches are suddenly free to grow, and the plant becomes much bushier. So, that perfectly illustrates the two main types of plant growth. Primary growth, which is driven by the apical meristem, is all about getting longer. But woody plants don't just grow taller, they actually need to grow wider to support themselves. That's secondary growth. It's driven by lateral meristems, like the vascular cambium, and it increases the stem's thickness, which is exactly how we get wood and bark. And an amazing side effect of that secondary growth is the formation of annual rings. In the spring, when there's tons of water, the vascular cambium pumps out secondary xylem with large, really thin-walled cells. That's called early wood. But then in the fall, it starts producing denser, thicker-walled cells, known as late wood. This stark visual contrast creates a ring. By just looking at these rings, you can literally read a tree's exact age, along with the seasonal climate history it had to survive. It's basically a historical record. Section 3. Plant hormones and responses. Chemical messengers in action. Okay, we have 5 major plant hormones to talk about. To lock these in, let's use some simple memory tricks from our sources. First, auxin is for stretch. Just picture a heavy aux cable physically stretching a cell longer. Gibberellin is for taller. Think of someone jibber-jabbering nonstop, which drives dramatic nonstop stem growth. Cytokinin is for split. Cyto means cell, so imagine a copy machine spitting out copies, splitting and dividing cells to make more. Abscisic acid, or ABA, is for stop. Think apply brakes when the plant is stressed out, like during a drought. And lastly, ethylene is for sweet. It's the gas that actually ripens fruit. So just remember, stretch, taller, split, stop, and sweet. Let's look at one of those hormones in action with a classic response model called phototropism. When light hits just one side of a stem, the hormone auxin actually runs away from the light, shifting over to the shaded side. And remember our trick? Auxin causes cells to stretch. So the cells on the dark side elongate much more than the cells in the light, physically pushing and bending the entire plant toward the sun. It's such an elegant, totally localized response to the environment. Section 4. Misconception and formative check. Testing your knowledge. All right, let's clear up a really common point of confusion for biology students. Both auxin and gibberellin are heavily involved in growth, and they both cause cells to elongate. So how do you actually tell them apart? Well, the trick is looking at the scale of the job. Auxin is all about localized stretching, like a single stem bending toward a window. But if you're looking at a plant where the entire stem is just dramatically bolting upward, getting super tall with massive spaces between the leaves, that's the heavy lifting of gibberellin. Okay, pop quiz time. Which plant hormone is most directly responsible for fruit ripening? Take a quick second, think back to those memory tricks we just went over, and lock in your answer. If you picked B, ethylene, you nailed it. Ethylene is our sweet hormone. And what's absolutely wild about ethylene is that it's a gas. That means it diffuses right through the air. So a single ripe fruit releasing ethylene can actually signal the unripe fruits around it to hurry up and ripen too. That's exactly why tossing a ripe banana into a paper bag with some unripe avocados works like a charm. Section 5. Concise Concept Recap. Bringing it all together. Let's do a rapid-fire review of these grade 9 botanical concepts before we go. We started right at the cellular level. Meristematic tissues are the dividing engines, while permanent tissues, like those dead hollow xylem pipes and the living phloem, take on highly specialized transport roles. Zooming out, we explored primary growth at the apical tips for length, and secondary growth on the lateral sides to create thickness and wood. Finally, we covered the 5 chemical messengers running the whole show. Auxin to stretch, gibberellin to grow tall, cytokinin to split, ABA to stop during stress, and ethylene to ripen fruit. I'm going to leave you with a provocative thought to chew on. We know the environment shapes a plant, but think deeply about the internal signaling. If a tree goes through a brutal drought, it releases the stress hormone ABA to slam on the brakes and save water. So how might that invisible chemical signal physically alter the permanent history recorded in the dense, thick-walled cells of that year's annual ring? It is a perfect example of how microscopic chemical signals literally write macro-level history. Thanks so much for joining me on this explainer, and keep exploring.